Welcome to the Proteomics course. In today's lecture, we will talk about applications of cell-free protein microarrays. In previous lecture, we discussed how different type of cell-free expression system can be employed to generate protein microarrays. We discussed different type of approaches, including protein in situ arrays, nucleic acid programmable protein arrays, multiple spotting techniques, halo tag arrays, as well as DNA array to protein arrays. So, in general, protein microarray technology allows the identification and quantification of proteins in high throughput manner. The recent advances in the protein microarray technologies, they offer unique opportunities to find novel biomarkers as well as apply this platform for several applications. The combination of proteome scale technologies, especially the protein microarrays, have potential to apply for wide variety of biological applications. The application of cell-free expression based protein microarrays have seen a rampant increase because of the ease of synthesizing the proteins by using cell-free expression based system as compared to the cell based traditional way of purifying the protein and then printing on the array surface. So, in today's lecture, we will talk about an overview of protein microarray experiment. So, first of all, I will give you a broad overview how any type of protein microarray platform can be used for different type of applications. I will then describe few applications very briefly by taking some case studies. This will just give you an idea that how these platforms are used for wide variety of applications, including biomarker screening, immunological studies, as well as protein-protein interactions. Finally, we will touch upon the challenges of analyzing the microarray data, regardless of you are performing an experiment to identify the protein interactors or looking for new biomarkers. The microarray experimental setup, they provide very high throughput platform and generate data for the thousands of features simultaneously. Therefore, analyzing such data becomes very challenging. So, these 3 points will be discussed today. So, let us first start with an overview of protein microarray experiment. So, regardless of what application one wants to probe on the protein microarrays, there are certain steps, a workflow which is involved, which needs to be followed to perform such experiments. Now, protein microarrays, whether you have a cell based or a cell free platform, you need to perform certain steps. But if you are using the cell free based protein microarray setup, then you have to perform certain additional steps. The additional steps include synthesis of proteins on the chip first of all, and then all the steps regardless of it is cell based or cell free, it remains the same. It means in general, I will show you an overview of the various steps involved in performing a protein microarray based experiment. After watching this video and discussing about various steps which are involved in any protein microarray based experiment, let us now look some applications of cell-free based protein microarrays. Let us first discuss the biomarker identification. Biomarker discovery for the disease detection and pre-screening has been one of the major thrusts for the proteomics field. Biomarkers have potential to allow early disease detection as well as accurate diagnosis of the grade of the disease. These molecular signatures can also be used for the follow up of disease response, survival of patients as well as various other parameters. As you know, there is need for early detection and therapy of diseases such as cancer. However, the discovery of specific and sensitive molecular markers that remains challenging. So, people employ different type of technologies including protein microarray based systems to identify biomarkers which can be used for early disease detection as well as accurate diagnosis and different other applications. So, protein microarrays have greatly enhanced the biomarker discovery process because they allow a high throughput platform for simultaneous and rapid screening of thousands of protein. Many times the clinical samples are a limiting factor because you do not have a large amount of clinical sample to perform a study. In that regard also, the protein microarray is very impressive because with few microliter sample one can screen all the thousands of protein features simultaneously on the same platform. So, biomarkers have potential for early identification of disease state, monitoring a disease treatment response as well as follow up on the disease prognosis. So, let us look at how cell-free expression based protein microarrays have been employed for screening for the biomarkers. So, I am giving you the case study 1, detection of p53 autoantibodies using nucleic acid programmable protein arrays. The study is performed by Anderson et al. So, antibodies to several tumor antigens are identified in the breast cancer patient sera. However, there is very little knowledge about the specificity and the clinical significance of antibody immune repertoire in the breast cancer patients. So, Anderson et al. Adapted a specific detection of autoantibodies in breast cancer patients by using nucleic acid programmable protein arrays. The slide gives an overview of detection of p53 autoantibodies using NAPA microarray approach. So, let us discuss the details of this study in this animation. Biomarker identification. In this animation, we will discuss about detection of p53 autoantibodies in human serum using cell-free expression based NAPA microarrays. Study by Anderson et al. 2008. In this study, author generated protein microarrays based on NAPA expression. As you can see, in the NAPA chemistry, cDNA, BS3, BSA and capture antibody, these 4 features are printed on the chip surface as a master mix. After addition of the cell-free lysate, proteins are expressed, which can be then further probed with the diluted sera of breast cancer patients which contain p53 autoantibodies. In this study, detection was carried out by means of HRP linked anti-human IgG. The study detected p53 autoantibodies by means of NAPA microarrays, which was further confirmed by ELISA approach. As you can see, the spots are visible in the p53 positive sera, which are absent in the p53 negative sera. The p53 levels were found to be directly related to tumoral burden, with serum antibody concentrations decreasing after neoadjuvant chemotherapy. Now, let us talk about case study 2 for the biomarker screening. A bead based assay for multiplexed detection of antibodies to EBNA1 and p53, a study performed by Wong et al. So, Wong et al. Used a Luminex suspension bead array platform for the rapid detection of antibodies in sera. A programmable multiplexed immunoassay was used for the rapid monitoring of humoral immunity. As this slide shows the overview of the steps performed in this experiment, the author demonstrated that the method can be used for rapid conversion of open reading frame over a pheome derived cDNAs to multiplex bead ELISA for detection of antibody immunity in infectious diseases as well as for the tumor antigen identification. So, let us see the steps involved in this experiment by using this animation. We will now look at other application, rapid bead based assay for multiplexed detection of antibodies to EBNA1 and p53, study by Wong et al. 2009. In this study, authors developed a programmable multiplexed immunoassay where tagged antigens were expressed by using in vitro transcription and translation. And captured these onto the anti-tag coated beads. Once self re-expression step was completed, the synthesized proteins were further immobilized onto the beads through the capturing agents. These beads were then mixed together . After mixing the beads together, the serum was added to these coupled beads and human IgG were detected by probing with the enzyme linked anti-human IgG. The colored reaction was observed on addition of substrate to the enzyme. The authors demonstrated that this approach for detection of antibodies to Epstein-Barr virus nuclear antigen 1 or EBNA1 and p53. Let us now move on to other application, immunological studies. So, on one hand, there are several studies have shown application of protein microarrays, including cell-free based protein microarrays for biomarker screening. There are several studies have also focused on the immunological studies. This is case study 3, detection of potential immunogenic proteins of Plasmodium falciparum, a study performed by Doolan et al. So, Doolan et al. Used E. coli based cell-free in vitro transcription and translation based system to produce 250 Plasmodium falciparum proteins generated by the polymerase chain reaction and recombinational cloning approaches. After synthesizing the protein from these 250 ORFs, authors profiled antibodies that develop after natural or experimental infection or after the vaccination with the attenuated organism. Sera from malaria patients which have been exposed to the Plasmodium falciparum either naturally or experimentally were screened by using protein microarrays. In this study, authors identified 72 highly reactive Plasmodium falciparum antigens. The proteins expressed specifically in the pre-erythrocytic stage of Plasmodium, which was CSP, as well as some liver stage specific antigens such as LSA1 were identified successfully by applying cell-free expression based protein microarrays in this study. So, let us discuss this experiment and the study by looking at this animation. Let us now discuss the immunological studies in this animation. The use of cell-free expression based protein microarrays for detection of potential immunogenic proteins of Plasmodium falciparum was studied by Doolan et al. 2008. In this study, authors carried out cell-free expression of PCR amplified vectors using an Escherichia coli in vitro transcription and translation system. They expressed 250 putative proteins that were printed directly onto the microscopic array slides without any need for protein purification. These arrays were probed with serum samples from patients who had been naturally exposed to Plasmodium falciparum and who were experimentally exposed by means of radiation attenuated Plasmodium falciparum. Authors successfully identified 72 highly immunoreactive protein antigens as well as 56 previously uncharacterized antigens that were serodominant. The study has shown some of the newly identified targets can serve as potential vaccine targets. Let us now talk about case study 4, identification of immunogens of Q fever caused by Coxiella burnetii, a study performed by Baer et al. So, Q fever is widespread zoonosis disease caused by the Coxiella species. So, identification of immunogens of Q fever causing this disease were identified by using protein microarray based approach. So, in this study, authors used Coxiella burnetii protein microarrays to identify immunodominant antigens. Almost 2000 open reading frames were generated by using the cell-free expression based approach, E. coli IVTT system, and then employed this protein microarray platform for identifying the immunodominant antigens. So, some of the steps involved in this experiment will be discussed in the following animation. Case study 4, identification of immunogens of Q fever caused by Coxiella burnetii. Study by Baer et al. 2008. Baer et al. Carried out in vitro transcription and translation of 1988 open reading frame of C. burnetii by using E. coli based cell free systems. 75% of the open reading frames were successfully generated as full length proteins by using cell free expression system and then spotted onto the nitrocellulose arrays. These cell free expression based microarrays were probed with sera from the patients who had been vaccinated as well as acute Q fever patients. Fifty proteins were identified that were found to react strongly with the immune sera. So, as you have got a glimpse of different type of applications of protein microarrays such as biomarker discovery and immunological studies. Now, let us look at another widely used application, protein-protein interactions by using cell-free expression based protein microarrays. So, in this part of the lecture, I will mainly focus on the nucleic acid programmable protein arrays, how they have been employed to study the protein-protein interactions. In this slide, I am showing you a small test array which we used to teach a course in the Cold Spring Harbor in New York, where students were making these arrays themselves. As you can see, array layout, there are only handful 5 genes printed in the duplicate on the chip along with vector control, master mix and the water. Now, if we want to study the Jun and Phos protein interaction, and if we use the Phos as a query protein, the Phos query protein will bind with the Jun spot, and therefore, 2 spots are lighting up as you can see in the slide. So, in all the 6 blocks, there are duplicate of Jun proteins which are interacting with the Phos protein. So, Jun Phos interaction can be studied by using this system. Now, previous array was a small test array, but this slide shows you a high density array. Again, we want to test the protein-protein interaction of Jun Phos protein pairs. The students in this study used the Phos as a query protein, and then they identified Jun printed 4 times on the chip as the target. So, Jun Phos again was used for the model system to demonstrate how protein-protein interactions can be studied by using cell-free expression based NAPA microarrays. So, let us look at some case studies which have used the protein microarrays for studying the protein interactions. So, case study 5, identification of novel protein-protein interaction using nucleic acid programmable protein microarrays, a study by Ramachandran et al. 2004. So, Ramachandran et al. Reported generation of self-assembling microarrays, which was one of the novel technology reported in Science in 2004, and this study authors used a pairwise interaction among 29 human DNA replication initiation proteins, which recapitulated the regulation of CDT1 binding to the selected replication proteins and mapped its geminine binding domain by using nucleic acid programmable protein microarray approach. So, let me describe you some of the steps involved in this experiment by showing you this animation. Protein interaction studies. Case study 5, identification of novel protein-protein interactions using nucleic acid programmable protein microarrays. Study by Ramachandran et al. 2004. Ramachandran et al. Tested the use of NAPA microarrays by immobilizing 29 sequence verified human genes involved in the replication initiation on the array surface. And then expressing them in duplicate with rabbit reticulocyte lysate. The expressed proteins bound to the anti-GST antibodies, which are the capture antibodies present on the array surface. Authors made use of each of these expressed proteins to probe another duplicate array of the same 29 proteins, thereby generating a 29 by 29 protein interaction matrix. 110 interactions were detected between proteins of the replication initiation complex, of which 63 were previously undetected. Now, let us discuss the case study 6, high density NAPA array approach for studying well characterized gene pairs, study by Ramachandran et al. 2008. So, previous study was more proof of a concept where handful proteins were taken for studying the interactions, whereas this time a high density array was used where thousands of features were printed. So, Ramachandran et al. Used a high density NAPA approach to study the binary interactions between several well characterized interacting pairs such as Jun phos, P53 and MDM2. Now, selective binding to these interactions were identified by using specific antibodies. So, in the protein interaction, one thing which becomes very tedious to test out protein interaction in both directions. For example, if one is testing the Jun and phos interaction, it should work in either way. For example, if phos is printed on the array, Jun should be able to bind if it is used as a query protein, or similarly, if Jun is printed on the array, then phos protein can be used as a query for showing the interaction. Many times these interactions become unidirectional. It becomes very tedious to show that interaction is working in either direction. But in this study, authors showed that the protein interaction of Jun phos can be shown in both the direction. Now, in addition to showing that protein expression and protein interaction works, one thing which is interesting from this study that a co-expression can be performed. For example, there is not even need to purify the protein which is used as a query protein. So, if you have a protein microarrays, features are printed on the chip and then you have generated the contents by using self-free expression based system. Now, you want to study the interaction. For that, you have to purify a protein and you use that as an interactor. Now, you can use either protein specific antibody to identify the interaction, or you can use the tag specific antibody for detecting the interaction. But in this study, authors used co-expression. It means the query proteins along with the arrayed proteins were expressed by using self-free expression system. So, there was no need to in fact purify the query protein as well. When the protein interaction study has to be performed, you take the cDNA of phos, for example, mix it in the rabbit reticulocyte lysate along with the other in vitro transcription translation machinery, mix the whole cell lysate on the chip surface, and then after incubation, when proteins are expressed at the same time, the query cDNA will also express the protein, and then if it finds its binding partner, it is going to bind to those features which can be detected by using protein specific or tag specific antibodies. So, by performing this type of approach, authors allowed co-expression. It means same environment for both query and the target proteins. Now, both proteins are expressed in the same mammalian environment, and there is good likelihood that they are going to identify the right interactors. So, let me show you the steps involved in this study by showing you this animation. High density NAPA approach for studying well characterized gene pairs, study by Ramachandran et al. 2008. In this study, authors made use of high density nucleic acid programmable protein arrays to study protein-protein interactions. 647 unique genes were printed onto the array surface and expressed by adding the cell-free expression based system. After addition of cell-free expression based system, proteins containing GST tag were synthesized and bound onto the capture antibody. cDNA of the query protein was also added to the same mixture such that the query was co-expressed but remained unbound due to the lack of a tag capturing agent. These protein microarrays were then probed with antibodies specific to the query proteins. Authors detected various protein interactions using well-known query proteins such as Jun, FOS and MDM2. So, now we have seen the overview of how protein microarray experiments can be performed. We have looked at various applications by employing cell-free expression based protein microarrays. We have discussed biomarker screening, immunological studies and protein-protein interactions. Now, regardless of what application you want to perform on these arrays, you are going to generate large amount of data. So, the volume of data generated from the microarray experiment are prodigious. It becomes important to develop the appropriate informatics system so that one can analyze this data uniformly and make some very good output from this whole analysis. What are the major challenges of the microarray data analysis? Also, I'd like to get your comments that what should be the good statistical design when biologists are starting some experiment for the microarray, because most of the time these are clinical samples and they would like to get some very useful biological information from this.
Statistical is a very important aspects of these biological experiments, and I think the statistician gets involved from the beginning of the experiments. And the importance of statistician is, as you mentioned, about the sample, not only just sample size, but also to understand the experiment and control the variation in any biological experiment. And so it's a very good idea to have a statistician from the beginning of the experiment, and where they can contribute not only on the data analysis point of, but also to conducting and performing a optimal design experiment way, so you can have a as precise and as useful information out of the data or the experiment you are trying to achieve.
That's very important. But there are many ways of analyzing microarray data, and what are the different approaches which are available, and which one would you consider as a good approach?
Yes, there are statistical methods are tools, and it depends on your objective. So whenever you're trying to perform an experiment, I think one has to be very clear with the objectives, and which when you talk to the statistician, they will let you know what methods are appropriate your experiment to your hypothesis. And in that case, statistician will let you know how you should go and perform the experiment, and not only experiment but also to control the biological or technical errors which are involved in when you are performing experiment. So, but the methods, as I said, there are these new methods are evolving each and every day in this field, and it's because biological problems are very complex. It's not like you can answer by telling one method. So, but of course, there are some standard methods being used in other fields, and people are trying to use those like same methods in a biological point of view. Or I would like to say all the methodologies developed in statistics being based on the small sample size. And now in the recent years, so I would say nowadays we are dealing with a huge data set, and particularly in biological field. So in that case, we have to come up with new methods or new methodology in statistics which can handle more appropriately and more objective-oriented statistical method.
So I am convinced that it's not possible to really list out one best method.
Yes.
But at least can you provide a few possible solutions?
Absolutely. I will totally agree with you on that. And there is no unique method, and it's not only in biological point of, but even in journal as well, because statistical is always dependent, model dependent, or the experiment dependent techniques. So it only develops type of data or the experiment objective you're trying to achieve.
Sudesh, in the microarray field, biologists apply that to identify differential expression of genes. And what type of issues you see, like in terms of analyzing these data sets from the microarrays, and what are different ways of analyzing that data, and any comments on that?
I would like to say the statistical method used in identifying differential expression gene analysis, it starts from experimental design till the end of the analysis for till the final conclusion of the analysis. So there are a number of methods available, and particularly at design stage, because it's a design stage, there is a different kind of design. Like you must have heard about the loop design, reference designs, and also the factorial design. Is depending on your situation, you try to perform your experiment, and all this experimental design are basically depend on the statistical tool you want to use for your data analysis. And like when you are using reference design or you're using loop design, so basically you're trying to compare the two treatments, or the control, or the tumor. So in that case, you use t-test, like when you are comparing just two conditions. And in case if you are dealing more than two conditions, then you go a little bit analysis of variance kind of analysis, and then you use bootstrap method, you use SAM's method, and also there's another method called Welch method. So these are all methods appropriate in your situation when you are dealing with identifying differential expression genes.
So there are a lot of options available, but it always becomes challenging to apply which one is a real good one.
Oh, yeah, absolutely.
And that's why we need some good statistical people in the team for analysis.
I'm glad to hear that word, because most of the times what happen, the scientists or basic scientist research, they go to the statistician when they're done with their experiment, and they take their data to the statistician and they ask them, oh, could you please analyze the data? And so in that case, I would like to say there's a very famous saying from R.A. Fisher, when you go to the statistician with your data, so only the statistician can do a post-mortem of your data and can tell you how the data dies.
Very rightly said.
So with that remark, I would like to say, if you're trying to conduct any research hypothesis, or trying to perform any research-oriented biological question, so I would say go to the statistician from the beginning, and they can help you to at least get the optimal way of experiment itself, so that that way you can do the best analysis or best mathematics tools to appropriate your situation. So I would say, yes, statistician should be or must be involved from the beginning of the experiment.
So I must say that this is a take home from this interview, that it's not at the end, but actually from the beginning, when we need to involve a statistician for the large set of analysis if we want to perform some different high throughput experiments. And especially in the genomics and proteomics, is very important, because we invest a lot of technology in a lot of samples, and if our experimental design is not very well, then later on everything will fail. So with that thought, I will conclude this interview, and I'd like to thank Dr. Sudesh for being with us, and sharing some of his experience on microarray data analysis and challenges. Thank you very much.
Thank you.
So, in the image analysis, when you are talking about high density approaches, the data analysis, image analysis becomes very challenging. For example, you can see an image here for the protein microarrays, and as shown in this spot, the expression of this particular immunogenic protein is so high that it is spilling over to the neighboring proteins. Now, one need to correct for this or remove those spots which are in the peripheral of this protein. So, scaling up is good approach because one wants to perform high throughput experiment so that thousands of features can be studied simultaneously. However, while scaling up, especially when you are using the cell-free expression based approaches, one need to be cautious that what should be the optimum density for these arrays, because if there is a spillover of expressed protein on the neighboring spot, that is going to affect the values for the neighboring spot. Again, this slide shows how protein is diffused in the neighboring spot, which should be neighboring spot should be removed. Similarly, the background correction and various other parameters one need to analyze to perform a good microarray data analysis. So, let us have a discussion with Dr. Sudhesh Srivastava on microarray data analysis, what are the challenges involved, and then in subsequent lectures we will talk in more detail about different steps and nitty-gritties of those which one need to follow up for data analysis of microarray based systems. So, in summary, the protein microarrays offer novel technology for the simultaneous and rapid analysis of multiple biomarkers or interactors in high throughput manner. Microarrays have been widely used for detection of antigens as well as antibodies in blood sample and various other clinical samples. However, the traditional cell based approaches have certain limitations. Therefore, the cell-free expression based protein microarrays have emerged, and very strongly it has been shown that various applications can be performed without need to purify the proteins, because you can generate the protein content in the cell-free manner. In today's lecture, I gave you an overview of steps involved in performing a protein microarray experiment, whether you want to do the protein interaction or any type of disease screening. One has to go through all those steps. Then we looked at different type of applications in different case studies. Obviously, the case study discussion was very brief, but you can refer to those references and read those papers for further details. Finally, we got a discussion with a leading expert, Dr. Sudhesh Srivastava, on the microarray data analysis, and we will continue our discussion on applications as well as microarray data analysis challenges in the subsequent lectures. Thank you.